All right, I think we're good.
I
All
think
right,
so.
awesome. All right, hi everyone. Thank you for coming today. My name is Mansour. I am a biology major. I am working under Professor Meng Chen in the Department of Body and Plant Sciences, and our investigation is the identification of novel genetic elements in plant temperature sensing. So, let me go to slideshow. Sorry. So why is this important, right? So the big picture of this presentation is basically that these global warming is leading to ever-changing climates, right? Climates are volatile, they're dynamic, and we're seeing things that we haven't seen happen in a very long time. This is posing a significant risk to crop productivity, especially since a lot of our stable crops are based on these climates that have been relatively stable for the past few hundred or thousand years. So we're facing like this basically event wherein our climate is changing too rapidly for our food sources to adapt. And we need to understand plant temperature sensitivity and signaling responses so that we can actually predict how they're going to respond to these changing climates. And in the future, hopefully we can engineer crops and food staples that will actually be resistant to the stress that's going to come off these climates, because otherwise, for example, a large part of the world's population is dependent on rice. And rising temperatures are placing jeopardy on rice wetlands, right? So if we can understand more about these temperature responses, we could perhaps engineer rice that is going to be more temperature resistant, just as an example. So basically, we need to understand how plants recognize and respond to temperature. And our focus for this investigation is a model organism known as Arabidopsis thaliana. Arabidopsis thaliana is a common model organism for plant biology because of short generation time, has prolific seed production, small size, and it has very easy transgenic modification. It's basically the E. coli of plant biology. It's very ubiquitous in plant biology experiments. So our first question we have to address as a background is, what is thermomorphogenesis, right? The simplest way to define thermomorphogenesis is by its phenotypic effect, basically what it looks like to the common observer, right? A very simple way to simply say it is that when temperatures are warmer, you'll be exposed to hypocotyl elongation, which means your seedling will appear longer. And in cold temperatures, you won't exhibit this response, right? In the presence of heat, the seedling hypocotyl elongates. The reason for this, we believe, is that, or is commonly accepted, is that the seedling wants to have its leaves avoid the hot ground so that it doesn't evaporate the moisture stores inside those leaves, because if those leaves dry out, it'll severely jeopardize the seedling survival going forward. So what is the LRB gene family, and what does it have to do with thermomorphogenesis? So the LRB gene family are basically E3 ligases in the ubiquination pathway. Those are a lot of big words, so let me use this diagram on the left to try and kind of explain what's going on, right? So there's this thing called phytochrome B inside the plants, and in the presence of light, and in this specific example, red light, it translocates from the cytosol into the nucleus and is binded by these things called PPKs, which are photoregulatory protein kinases, and this thing called PIF3, which stands for phytochrome interacting factor 3, because it interacts with the phytochrome, and it's the third one. And it generates this complex here, right? And what we're interested in is this protein called LRB2. It's one of the 3 LRB proteins, LRB1, 2, and 3. They're all coded by 3 separate genes in this LRB gene family. And what they basically do is that they ligate a marker onto both phytochrome attracting factor 3 and phytochrome B. This marker actually marks these things for degradation, and the effect of this complex is neutralized by the presence of LRB, right? We don't know what modulates this protein, but we do know, based on previous observations, that plants with triple mutant knockout LRB123 do not respond to warm temperature and are temperature insensitive. Thermomorphogenesis is compromised in LRB1, 2, and 3 mutants, meaning that all 3 genes in the gene family are knocked out, and none of the 3 proteins are manufactured. So, and based on the graph on the right, you can see that the relative response is based on comparing the response at 21 degrees to the response at 27 degrees Celsius. You can see that the relative response for example, Columbia 0, which is our wild type control, and another mutant, phytochrome attracting factor 4-2, still have, even though its relative response is much less than your wild type, it still has a response, right? LRB123 has almost zero temperature response, whereas in these plants with all three of these genes knocked out will actually be nearly identical whether you grow them at 21 degrees or 27 degrees. So we know that temperature sensitivity fails when LRB is mutated. So what is LRB's role in thermomorphogenesis? This is our main question, really. And the approach we're going to have to answer that question is what we call a suppressor screen. So since this is emerging research, we haven't actually finished this approach, but we've gone through most of it. The way that we start a suppressor screen, which we've done, is we exposed these LRB123 mutant plants, our background mutation, to a mutagen, right? And then we screen for the phenotype, we identify possible candidates, and then we do a bunch of confirmations to make sure we actually have what we're looking for. For example, we screened at 27 degrees Celsius, we've done that. We identified possible candidates, we confirmed the background by genotyping, and then we did another actual temperature screen to make sure that we actually have plants that are temperature sensitive, not just plants that are tall. I'll explain that in more detail later. And then our future going forward, you know, we're going to have to backcross these to our original parent mutation and then try to sequence and actually see what this suppressor is. The whole point of the suppressor is to find a mutation that is involved in the pathway of temperature sensitivity, because if this mutation can rescue temperature sensitivity in a plant that does have mutated LRB, then we can discover other novel genetic elements involved in temperature sensitivity. So things we've done, right? We've done mutagenesis using ethyl methanosulfate, commonly referred to as EMS. We harvested about 1,500 EMS mutant lines, which we now refer to as suppressor of LRB123 in thermomorphogenesis, SLT. This is a result of our confirmations. So the first batch of phenotypic confirmation was screening 1,000 of these SLT mutant lines for temperature sensitivity at 27 degrees Celsius to just see if they have a response to warm temperature for the sake of efficiency. And this is an example of candidate isolation, right? This is what we do. So we have our wild type in the upper left corner, Columbia 0. And as you can see, the Columbia 0 plants have long seedlings. And in the bottom right corner, we have our negative control, LRB123, which have very short seedlings. You almost cannot see the hypocotyl, whereas in Colo O, as we refer to Columbia 0, it's a very long and stark hypocotyl. In our other candidate lines, I've circled in red examples of plants that exhibit long or extremely long hypocotyls in response to being grown at 27 degrees. So the examples of temperature sensitivity. And here's just a few more for 2 more lines. So this is just a graph to characterize our isolated candidates for our first run of phenotypic confirmations. As you can see, we have our controls up here, our positive and our negative control. Columbia 0 is very tall, and LRB123 is very short. And for our other suppressor candidates, we have plants that range from tall, medium tall, extremely tall, low, medium tall, which is just taller than short, really. And some of them are very tall, actually, even taller than Columbia 0. And these ratios are basically how many we manage to find out of the amount of seeds, for example, we plated on these plates. Some of the reasons the ratios aren't exactly, very high is probably because a lot of these seeds, for example, some seeds don't germinate. There's a whole bunch of other factors that go into it, but this is just the first rough run to just see what possible candidates we have. So after confirming genetic backgrounds in those candidates, we then take the 11, which both pass the phenotypic and genotypic confirmations, and we screen them at both 21 degrees and 27 degrees. Before, we only screened them at 27 degrees because screening over 1,000 lines at 2 different temperatures would be very time consuming and very resource intensive. But this way we can identify plants that are possible temperature sensitivity. The point of this run is to make sure that these plants aren't just for example, just naturally tall, right? Even if they have the background mutation, they may just have another mutation that makes their hypocaudal elongated. We need to make sure that their hypocaudal length actually responds to temperature. And so this is the result of temperature sensitivity confirmation, right? So as you can see that Columbia 0, our positive control, is short at 21 degrees and tall at 27 degrees. LRB 123 is short and short. And then these other plants, as you can see here, are short and then taller at the higher temperature. And the ratio at which they're taller is also something we keep in mind. For example, SLT 150-22 and -24 exhibit almost 100% of their plants to show this temperature sensitivity phenotype, whereas 25, 28, and 29 exhibit about 75% of them showing that temperature sensitivity phenotype. This is already where we start thinking about our future plans of determining if our mutation is dominant or recessive, because we know that if it's dominant, there's always a chance you have a phenotype that passes for the dominant phenotype, but is actually heterozygous. So really, in reality, if 75% of your plants are showing that dominant phenotype, then maybe 25% of them aren't because they're recessive, right? They're little a. And then 50% of these could be big A, little a, and 25% of these could be big A, little a. But we won't know for sure until we run our backcross. Whereas on the other hand, these plants that show 100% phenotype are going to have both, most likely are just recessive and have little a. And we graph the responses below. So these SLT 150 lines, as always, have a significantly greater response than the negative control, but it's also much weaker than the positive control. But the point really is to just make sure that we find plants that rescue temperature sensitivity, not the intensity in which they rescue it. The 296 lines are also not very strong in their response, but it is still statistically significant when compared to our negative control. So future plans is to continue the suppressor screen procedure I previously mentioned, which is we're going to backcross them with the background to determine once and for all if our mutations are recessive or dominant. And based on that, we're going to generate a V2 mapping population, which we can use to isolate DNA for Illumina sequencing and then identify our causal mutation via a bioinformatics pathway called SIMPLE. So at this point, I'd like to give thanks to everyone involved in the project, the postdoctoral researcher who basically held my hand the entire way, Jiangwen He, my principal investigator or my PI, Professor Meng Chen, the Plastic Genome Lab, which is the name of our lab, and the UCR Department of Botany and Plant Sciences, as well as UCR student engagement, and everyone here for showing up today, and for the mini grant I received to fund a small portion of this project. Thank you, everybody. I'm going to show my citations now. I forgot the slide existed. And any questions? I'm going to stop recording.